I think it's important, again, I know I keep going on about it, but context of use, that is what is really important. And if you think back to the EMA slide where they're saying that the regulatory impact will dictate the level of qualification necessary. Obviously, if we're wanting clinical trial waivers in special populations, we have to have a reasonable amount of data to be able to support that in terms of its qualification. And obviously, there have been ongoing efforts over the last few years. And if we want to get regulatory adoption, we have to, what is good about this is the IQ publication that came out a couple of years ago, where they looked at PBPK modeling in renal and hepatic impairment. And the good thing about this was that there was a nice cross-section of drugs available from each of the industry members who contributed to this analysis. Also, from our own in-house consultancy case studies, we can continue to apply the models and gain experience there and submit them to the regulators as well, just to gauge their level of interest and to share these data. And also, where possible, we've been publishing this data as well to put it in the public domain. But on top of this as well, we recognize that there may be gaps, and therefore we have to continue advancing the science. And that is just indicated in this particular publication here. So just to quote, I think one of the FDA presenters from last week from the ISISX webinar, I thought it was quite interesting there because they were giving their position on where we are with respect to organ impairment and regulatory submissions. And I remember from one of the slides, I just saw verification, verification, verification. And I think, that is essentially what I'm saying here, is that there are emerging examples, independent analyses, and therefore we have to keep these coming out, but we also have to think about the best way of actually utilizing this data going forward if we want to be able to qualify the platform. And also, I think importantly as well, if we're wanting to get regulatory adoption as well, you'll be aware that the renal impairment guidance has been undergoing review and been circulated to everyone. And therefore, I think it's important to think about, based on the data that are available in the public domain, what are the approaches that we can use? What are we more confident in? And this was just a publication that a colleague and I put together, and just thought process as a perspective in CPT, where we were thinking that, based on our experience, we've seen a number of the, examples or case studies where we've got data available in severe renal impairment or severe hepatic impairment. And if you have those data available to verify the, the model, then perhaps we can extrapolate to untested scenarios, perhaps in mild renal impairment, and also following multiple dosing as well. So this was just a schematic that we put together. And I think, these types of analyses are important going forward, and especially when we're wanting to look at all of the different special populations and thinking about the best way that we can actually, provide support for regulatory submissions. So without further ado, I just wanted to acknowledge, apart from the individuals who were on the publication, the CYP qualification publication, I just wanted to acknowledge the, the efforts of the Simcyp team, because, this is not just based on, work from 6 individuals, but from many years of work. And I just wanted to acknowledge their contribution. So I'd like to thank you very much for your, for your interest and attendance. And I know that qualification isn't the most exciting topic available, but we felt it important to share the findings of our study, and also to indicate that we are, trying to move things forward in this particular area. Thank you very much.
Thanks, Karen. So we're going to move into a period of questions. So just a reminder, you can use the Q&A functionality to send your questions through. I'll kick us off with some questions. Why do we need qualification if submissions are already approved?
So I think, that's a really interesting question, and in fact, a very important one. And, and it's something that really I should have addressed during the presentation. But You know, just to go back to the CYP3A4 example, or CYP DDIs, you know, I think many people would say, why are we bothering to qualify, you know, competitive inhibition for CYP enzymes when there are so many examples out there? And I think the issue that we have here is that, you know, depending on which regulator it goes to, and within the EMA, obviously there are a number of different agencies, and you could get, you know, a very different outcome depending on where it ended up. And in terms of the formal qualification, what we're talking about here is having a standardized approach. And I think this is where the EMA are going with this. So in other words, if we submit qualification for CYP DDIs for competitive inhibition across the board, then at least it will take away that subjective, I think, viewpoint with respect to looking at the PBPK models, and hopefully a more streamlined process, and yes, and more transparent as well.
Yeah, makes sense. Thanks. Just a note, because you mentioned in the presentation that you were using version 19. Obviously, that's a few versions now. We're on version 21 in the simulator. So is regulatory qualification version specific? Do you have to do every time?
So again, you know, yes, I did mention during the, you know, during the webinar that, you know, the publication that we did, all of the work was done in version 19, because of course that's what we had available at the time when the work was initiated. And of course, this is quite typical of, you know, many of the submissions done with a consortium. They would have started with the version that was available when they did the initial model development. And so I think going forward, you know, to answer your question, the qualification is version specific, but what we have been doing, and what I've seen by some of the regulators, is that even if you provide supporting qualification for a different version to that was, you know, used for the investigational drug simulations, I think as long as you can show version control and that there's been little change across the versions, and you provide supporting documentation indicating that, say, for example, the compound file summaries, then it can be extrapolated to other versions, the findings. Yeah.
Okay. Yeah, we had a similar question as well, basically saying, if you had to go through it with every version, does it really pay off? Anyway, so another question is about complex DDIs with PBPK qualification, with more than one mechanism for inhibition or induction for perpetrators, and especially for substrates. So, yeah, can you comment on the process involved for qualification in these circumstances?
Yeah, so again, it goes back to, you know, finding relevant examples. So, you know, obviously I've simplified it somewhat by talking about the processes independently, but I think we're collecting enough examples now of where we've got transporters. We've also got UGTs and also CYP enzymes. And for those examples, it will be important to utilize, I think, published data or data that have been made available in the FDA reviews, collate those examples, and essentially put them together in a single document to support those. But we are seeing an increasing number of examples. But again, you know, I mean, going back to the point that I was trying to make earlier, I don't know what the magic number is in terms of the, you know, amount of, you know, number of examples for qualification, because I've seen in some cases, you know, where, you know, for CYP3A4, we've had a lot of supporting data. I've also seen instances where there've only been a handful of examples. So, you know, again, there are a lot of different examples out there, but it's a case of pulling them together. And that's why, as I said, I think it's interesting in terms of, it was interesting for us to see in terms of priorities.
Yeah, I think this kind of leads on to what you're just saying in terms of priorities, but obviously we've got a long history of using PBPK for CYPs, especially as we've talked about today, interactions, and lots of data for verification. But given the large data gaps, are we really ready for qualification, for example, in special populations?
So, again, I go back to the point that I made, about the FDA presenter from last week in the ISSX workshop, verification, verification, verification. I think, and it comes back to, again, what level of qualification we want to, we provide. Because obviously, when you do submissions to the, to the FDA, they are asking for, qualification or examples showing that, the platform works sufficiently well in those areas. And that was what was mentioned with respect to special populations. So it may be that we don't have enough data, I think, to go for formal qualification and endorse the platform across the board for the special populations. But I think, there are enough examples available in the literature or emerging examples that can be collated and put together in the form of a document.
Okay, great. Related to populations, there's a question here about if we change some parameters in populations, for example, gastric emptying time, should we re-verify Simcyp default compound files, midazolam, itraconazole, etc.?
So I would recommend looking at doing that, yes. If you're using those compound files for the support in that particular population. So I'm assuming that what you mean there is that if you change, say, for example, you were looking at renal impairment and you run a simulation and you decided to change the gastric emptying there, would you then do the same for the other? Yes, I would. I would look at both scenarios. Yeah.
Okay, great. There's a comment here about the 10-fold sensitivity analysis for Ki for a competitive inhibitor. The comment is, it seems like it's a lot if you're working with high quality in vitro data. Was there any consideration that the 10-fold adjustment needed in some of the PBPK models for inhibitors came out of lack of solid in vitro data across various compounds, or a good understanding of FU-MIC?
So, it's always, so that's a great question, and one that we've, the analysis we've taken apart many times over, because at the end of the day, when you're doing a meta-analysis, what is the best way of doing it? Do you put all of the data available there, correct for non-specific microsomal binding, and then just take an average Ki value? Or do you streamline them, look at them to see which ones, certainly in my view, would have robust methodology and had corrected and used low protein concentrations? And so, we've had a look at all of that. And so you say that 10-fold seems quite high. The problem that we have is obviously across the enzymes, when you look at the spread of the in vitro Ki values, in some cases it's much higher than that. And that is why what we've done is recommended that, or tried to say for CYP3A4, that we're pretty confident that the in vitro Ki values hold, but then we've provided the criteria that, your positive control Ki value should be similar to one that we've used in the, the analysis that we provided. And I think, for the other CYP enzymes, probably what we could do going forward, as we increase the number of examples that we have, we would be able to provide similar guidance for that. But at the moment, the 10-fold is the best that we can do based on that data.
Yeah. Okay. Thanks. So in terms of access to post-marketing surveillance data to verify PBPK models, do we have access to that? For example, if the model predicts no DDI and the clinical trials are waived, do we see the same in the real-world clinical data? Maybe do you have any examples of
that? No, I don't. I don't have any examples of that I can share with you today, but I think, this is the point really that I'm trying to make, during this session today, is that there are a lot of data out there. It's a case of deciding, what, so there are several things here that we need to think about. There are a lot of data available, but what type of data do we need, and what is the best data that is actually going to help us qualify the platform going forward? And so I think that's, a very key point. But I think if people do have examples that they can share with us, then it can only, help us, build better qualification examples and reports for submissions.
Mm-hmm. good. I'll just quickly go back to the tenfold Ki question, if that's all right. And it was just a clarification on whether it does tenfold Ki for 3A apply to both gut and liver. Was FU gut assumed to be 1 in the analysis? I guess in the first question, yes, it's applied across the board, but I mean, the FU gut will be an interesting one.
Yes, and so what we tried to do was look at that with respect to FU gut, and of course what we have in Simcyp will be a mix of FU guts depending on the actual recovery. So at the moment it wouldn't be possible for me to answer that question, but what we can do is go back and do a sub-analysis and provide that information. Yeah.
Mm-hmm. So there's a comment here from Neil Parrott, and he says there are 3 possible applications: induction, transporter, and hepatic impairment. I guess that was the organ impairment options that we gave in the polls. The hepatic impairment might have the biggest impact, but faces big challenges given the complexity. Do we have the right data and understanding of hepatic pathophysiology to make progress at the moment, or is a more long-term effort necessary? And if any plans at Simcyp for such work?
Oh, well, I'm going to bat that one to you, Ollie.
Yeah. So, well, yeah, I can say for organ impairment, that is something that, as Karen's mentioned, that we're starting to do. In terms of making progress with the hepatic impairment populations, we obviously made some strides, I think, with version 21 and trying to incorporate other features within the model. But I think even from what was presented even last week at ISSX, it was really interesting talk by Tycho talking about the impact on absorption and bile salts and things. So there's still lots of work to do for the hepatic impairment populations, I would say, but it's definitely something that we are continually working on.
Yes, I think just to add to that, you know, I think also what came out of the ISSX, you know, workshop presentation last week was that at the moment, I think the level of the confidence in hepatic impairment models is such that they would request clinical studies in each of the, you know, mild, moderate, and severe. But I think, you know, there's still the utility for PBPK going forward, and, you know, for multiple dosing and also potentially DDIs as well. And so that goes back to my point, is that I think even if we feel that we're at the position where we can't avoid clinical studies, we still have to then provide supporting documentation, I think, to support all of these other scenarios.
So there's quite a lot of interest in the Ki, and there's one more question here. Have you observed in your analysis any bias by system, i.e. Ki from HLMs or recombinant CYPs?
No.
No. Okay. So see if there's any more questions. I think we're about at time anyway. Yeah, if there's any more questions that come in, we'll try and answer those ones. But I think in the interest of time, and it's hit top of the hour, I think we should close this. But before we go, just to mention that there is an upcoming webinar on the 22nd of June. This is from Emi Kimoto and Metaanalysis Pharma, and they're looking at OATP1B mediated DDIs, and also quite a hot topic at the moment, looking at biomarker informed model-based risk assessment. So please register for that one on the Simcyp members area if you're able to do that. And then finally, I just wanted to again thank Karen for your time and giving the presentation today. So Thank you for that, and thank you for everyone for joining online.